Hi everyone, welcome back to this channel. So today we are doing biochemistry one-shot revision, and I would like to keep this video as short as possible. So without delay, let's get started. So I will start with sugars. So there is a term named reducing sugars. So mostly monosaccharides are reducing sugars. Monosaccharides means there is a single unit, but there are disaccharides which behaves as reducing sugars. So examples of disaccharides that are reducing in nature, that is lactose, maltose, lactulose, and cellobiose. And if there are more than 2 units in a particular saccharide, then that polysaccharide that is termed as polysaccharide, and they are all of two types, homosaccharides and heteropolysaccharides, right? So common example of homopolysaccharides are starch. It is made up of D-glucose units, and amylose has alpha 1,4 linkage. This is a like linkage, glycosidic linkage. And the amylopectin, it has alpha 1 to 6 linkage. Let's quickly talk about glycogen as well. This is indeed made up of D-glucose units. It also has two linkages, alpha 1 to 4 linkage and alpha 1 to 6 linkage. Okay. There is one more thing, that is cellulose. This is also made up of glucose, but it has only beta 1 to 4 linkage, that too in transconformation. Okay. Now let's quickly talk about lipids. So there are many lipids. Let's say 12 carbon is lauric acid, myristic acid is 14 carbon units, 16 carbon units is palmitic acid. So basically there are two kinds of lipids: saturated lipids, that is fatty acids, saturated fatty acids, and unsaturated fatty acids. But the saturated fatty acids, they have high Tm value, that is melting temperature. So there is a relation between Tm with chain length and unsaturation. So as the unsaturation in a particular fatty acid chain increases, the Tm value decreases, and it is directly proportional to the chain length. If the length of particular fatty acid chain is more, then its Tm value will be high. Okay. Now I would like to proceed for amino acid thing because it is most commonly asked. So general structure of amino acid have a carboxylic group, a amino group, and a normal hydrogen, and a R group. This R group is variable. Different kind of R groups will come, and different type of amino acids will be found, right? So based on this variability in R group, there are different kinds of amino acid: acidic, hydrophobic, and several other types. But here I would like to teach you about pKa values. So pKa means the pH at which a particular group or ion, this particular side chain molecule will become dissociated. If I talk about this NH2, it will gain a hydrogen and will become protonated, right? But if I talk about this carboxylic stuff, it will be deprotonated. Great. So there is a pH at which this reaction will be favored. There is a pH at which this reaction will be favored. Similarly, these reactions exist for R groups as well. So we need to remember this because we have to, if they give you a question of a peptide that you have to calculate the charge on the peptide. Then these things will be helpful there. Okay, so for the carboxylic group, the pKa value is 2.2, ideally between 1.8 to 2.3. I would like to tell you one more thing. If the pH of surrounding environment is greater than pKa, there will be zero charge on the amino acid. But if the pH is greater than pKa, then the amino acid will possess negative charge. And if the pH is less than pKa, the amino acid will possess positive charge. So for amino group, the pKa value is 9.8. You can also approximate it to 9.5 to 10.5. For pKa value of SH group of cysteine, it is 8.3. For asparagine ka R group, it's 3.65. For I'll just write it down quickly. Okay, so you just remember these values because this is going to help you for the amino acid ka charge calculation as well as for the peptide calculation. Okay, so I would like to tell you that there is a relationship between this pK value and the acidic strength. If the pK value is greater, the acidic strength will be lower, and if it is higher, then the basic strength will be higher. Okay. Just to remember that glycine acts as a inhibitory neurotransmitter. Let me tell you about this glutamic acid. Glutamic acid acts as an excitatory neurotransmitter, but when it is in form of GABA, it acts as inhibitory neurotransmitter. Okay. So earlier we talked reducing and non-reducing sugars. So what it's based on. So if a particular sugar, let's say there is a solution named Benedict's reagent. It basically has copper ions in it. So if this is being converted to sodium citrate, like not only copper ions, like complex is there. So if this is the case, then the solution will turn red brick color. If this is so, then we'll conclude that the sugar is reducing. Great. Let me tell you quickly about a particular thing, that is the Ramachandran plot. I mean, it's just a revision, right? So there are 5 angles in this. There are psi angles, minus 180 to plus 180. So in this particular region, there are left-handed helix, alpha helix, and in their anti-parallel beta sheets, and here parallel beta sheets. So basically you can just remember this. So if there will be options, you know the range, right? It is between like the psi angle is between 0 to plus 180. So if some value is between this, then you can say if the psi and phi angles are this, then the particular structure is parallel beta sheets or anti-parallel or left-handed helix. So in this particular range, there are right-handed alpha helices. Now, There are many factors that affect the protein structures. For example, the pH. It affects the hydrogen bonds, and heat. Heat affect all type of interactions. That's what we say, na, कि at high temperature protein become denatured. Means all interactions are being affected. There are organic solvents, urea, and detergent. These things break hydrophobic interactions. Okay, so just to remember, average molecular weight of a nucleotide is 330 gram per mole, and the average weight of an amino acid is 110 gram per mole. Okay, so if GC content is more in a particular DNA sequence, then its stability is more, and the TM value will also be more. Just to remember, if in a particular question they give 4 sequences and they ask which one of the following is more stable, then you have to check it for GC content. You should calculate the number of G's and C's in the given sequences, and then based on GC content you can conclude whether it is stable or not stable. Okay. So I will quickly tell you about bDNA. Diameter of bDNA is 20 Armstrong. The number of bases in helix. Ideally 10.5, but sometimes in the question it is given 10. So you can consider 10 as well. The helix height is 34 Armstrong, and the distance between two bases is 3.4 Armstrong. Number of twists, that is 10.5 per base. Okay. Now the next thing is enzyme kinetics. This is important. From this portion also I've seen many questions. So there is a term named Vmax, that is the maximum velocity of the reaction achieved by a particular enzyme, that is Kcat into Et, where this is the catalytic activity and this is the total enzyme concentration. And affinity of an enzyme towards any particular substrate is inversely proportional to the Km value, and also dissociation. So Kcat is the catalytic rate constant, or also you can say it turnover number, which means if the Kcat value is more, then the product formation will be more. For an ideal enzyme, we want more product to be formed, so the Kcat should be highest, but also we want maximum affinity, so the Km should be lowest. Okay. Now there is also inhibition, right? So the competitive inhibition. What happens in competitive inhibition is the Vmax remains unchanged, but the Km value increases. So this is sort of graph that looks like Lineweaver plot. This is straight, right? No inhibitor. This is in presence of inhibitor. For non-competitive inhibitor, Km remains unchanged, means affinity of the enzyme don't change, but the Vmax decreases. For uncompetitive, Km decreases, Vmax also decreases. Now let's talk about some of the endopeptidases, means the enzymes that cleave the particular peptide at a particular amino acid position. So the trypsin, it cuts from C side, carboxylic side, and it cuts at RK, that is arginine and lysine, but it don't cut proline. There is chymotrypsin. This also cuts from like carboxylic side, but on wife. Wife means tryptophan, tyrosine, and phenylalanine. It also do not cut proline. CNBR, this cut at methionine position from carboxylic side. It also don't cut proline. There is one more enzyme, that is pepsin, but this cuts from amino acid, amino side, amino group, this ammonia group. It cuts wife, means this amino acid, tyrosine, phenylalanine, and tryptophan. It do not cut proline. So for denaturation of disulfide bonds, DTT is used. Okay. Now let's talk about few of the formulas that are more important in terms of solving NAT questions numerical. So let's say enzyme and substrate with the K1 rate, they form enzyme substrate complex, right? And the backward reaction rate is minus K1. K2 with the K2 rate, the product is formed, right? So the Km, that is the affinity of the enzyme for the substrate, will be given by minus K1 plus K2 upon K1. What is it? That is dissociation by association. So this is associating the substrate with the enzyme, but these both rates are dissociating the enzyme from the substrate, right? And also there is another metric, that is catalytic efficiency. If somebody asks you to calculate catalytic efficiency, then you should just divide Kcat by Km. Okay. Because for ideal enzyme, the Kcat will be higher and Km will be lower, means the catalytic efficiency will be highest. Okay. There is another formula, that is Vmax is catalytic efficiency ka Kcat, and this is ET. It is a total enzyme concentration. And if you have to calculate the velocity of the particular reaction at a observed velocity, means then multiply Vmax, this is a substrate concentration, Km plus substrate concentration. But in case of competitive inhibition, this formula will become Vmax substrate concentration, Km will be changed, right? Vmax won't change, but Km will be changed. So 1 plus 1 by Ki. This is inhibitor concentration plus substrate concentration. Only Km is changing, right? but this Vmax is not changing, but the overall will change. Okay, so the next thing is, let's talk about solutions a bit. So if I have to find the percentage of solute in a particular solution, then what I will do, the weight of the solution, I mean the solute in the solution divided by the volume into 100. There is one more formula, C1V1 is equal to C2V2. This is the matrix for stock and this is for the working solutions. This is a concentration of the substrate in the stock solution, volume of the stock solution you are using to create your working solution, and this is the concentration of solute in the working solution and the volume of the working solution. Okay. If you have to find the pH of the solution, then what you have to do is negative log of hydrogen ion concentration, or the inverse of hydrogen ion concentration, take the log. There is important thing to note, if a pH changes by 1 unit, your hydrogen ion concentrations will change by tenfold because it's on the log scale. And the pH and pOH should be 14. pOH is negative log of OH ions concentration. pH of very dilute acid solutions, their pH will be slightly lesser than 7. And pH of like very dilute like basic solutions, their pH will be slightly more than 7. Okay. If you have to calculate the Ka, that is the dissociation constant, then just do hydrogen ion concentration, molar concentration. This is the conjugate base and this is the acid concentration. Then for pKa means negative log of Ka. If the pKa changes by 1 unit, the dissociation constant will change by tenfold times. And the pK value of acetic acid is 4.76. There is one more important equation, that is pH is equal to pKa plus log of conjugate base concentration divided by acid concentration, molar concentration. Okay. I have covered most of the important things in biochemistry. So here I would like to end this video. Best wishes for your exam. See you in the next video.